Support for Consider This is provided by
There's a new place for senior care with daily laundry service. A place with a day spa. A place where seniors get the care they need in the comfort of home. Home Instead Senior Care.
From the campus of the University of Nebraska at Omaha. Consider This, a program of issues and information behind today's news.
DNA is the instruction manual for how to build life. From animals to plants to humans, it defines us all. Information is stored in DNA using 4 types of molecules, which come in pairs. We have billions of these pairs. DNA folds itself into paired packages called chromosomes, which are stored in the nucleus of cells. Each species has a different number of chromosomes. Humans have 46 chromosomes, which contain our genes. Genes act as instructions for molecules called proteins. In humans, genes vary in size from a few hundred DNA bases to more than 2 million bases, and we have an estimated 20,000 to 25,000 of these genes passed during reproduction. Each person has 2 copies of each gene, one inherited from each parent, and most genes are the same in all people except for a small number of genes being slightly different. These small differences contribute to each person's unique physical characteristics, varying from height, color, and personality traits. Some genes are dominant and some are recessive. Dominant genes are more often expressed as observable characteristics, while recessive genes are often masked by dominant genes. The recessive genes are expressed when inherited from both parents. While we share 98% of our DNA with bonobos and chimpanzees, we don't quite look alike because that 2% equals thousands and thousands of different genes. And while all humans share over 99% of our DNA with each other, what we don't share is what makes us unique in our own way.
Hi everyone, I'm Kathy Wyatt. Welcome to this week's edition of Consider This. Our topic, DNA testing. Here to talk more about it are Dr. Omar Rahman, Director of Genetic Medicine and Friedland Professor for UNMC's Monroe Meyer Institute, and Jason Linder. Jason is Biology Section Manager and DNA Technical Leader for the Nebraska State Patrol Crime Laboratory. Thank you both for being here and welcome.
Thank you.
Okay, Dr. Rahman, we're going to start with you. For those of us who didn't quite excel in biology, right? Key points from that opening video.
You know, we've known for a long time that children look more like their parents than they do the average person in the population. And we know that because there's some type of information that's passed on from each parent to the child, and that each child represents a mix of mother and father. It wasn't until the 1950s that we understood that that information was carried in the form of DNA. And DNA is simply an instruction manual on how to build a human being. Half of your DNA comes from your father and half of your DNA comes from your mother. And the information is written in an alphabet, just like a book is written in an alphabet, of 4 letters, A, T, C, and G. And it's that combination of information that codes for different parts of the body. So what are the eyes going to look like? What is the hair going to look like? What's the height going to look like? When DNA was first discovered, the very first advances in understanding how DNA worked was really in looking at human disease. So the first genes to be identified were for common human genetic disorders like cystic fibrosis, sickle cell anemia, or muscular dystrophy. But the genes themselves only comprise maybe 1% of all of the DNA in your body. So as the technology has advanced, we've now begun to look at the other 99% that make up our DNA. And we're beginning to realize that there's not clear definitions of health and disease, that everything is kind of on a spectrum. And where you fall in that is really a mixture of the genetic material you inherit and the environment that you develop in.
Okay, which leads us into DNA testing. Talk a little bit about that.
So in the past, DNA testing was a very painstaking process. You often, as a clinician, when I see a patient who might have a genetic disorder, you often had to know what genes you were looking for and go after those specific conditions. With some advancements, probably about 10 years ago, in the way DNA is sequenced, we now have the ability of sequencing all of that those letters within the genetic material very, very rapidly. There's about 6 billion of those letters in a single cell. And so if you look at in 2000, when the Human Genome Project was first completed, it took 10 years and nearly $100 million to do a single person's genome. And we now can do that in a matter of hours at less than $1,000 per person.
Wow. This has consumed a good chunk of your professional career.
Mm-hmm.
Why?
So it's incredible to think that where we worked as in terms of clinical genetics years ago, we were very focused on rare diseases. And now we realize that almost every condition is genetic in origin. So heart disease, cancer, diabetes, obesity, autism, all of those conditions have genetic contributions. They're not purely genetic, like cystic fibrosis or sickle cell anemia are, but it's definitely an interplay between your genetics and your environment. And we now, with the advancements in DNA testing, can really begin looking at a person's genetics and their environment and begin to make assumptions about what are their risks for these common multifactorial conditions.
Talk a little bit about your current opportunity to apply all of that knowledge.
So I think the most exciting thing in our field is in the area of precision medicine. If we can understand the genetic contribution of a particular disorder, then maybe we can use a person's genetic information to tailor treatments for them. So in the past, for example, when we would treat hypertension, you would take 100 people and you would give them all the very same first-line drug, and maybe half of them would respond and half of them wouldn't, and then you would have to adjust the dosing accordingly or change a different drug. And I think every person who's ever been to a physician knows that. You get put on a typical first-line drug. Nobody is actually looking at what's unique about you that maybe we can tailor a therapy to get you on the right drug very soon. That area is actually called pharmacogenetics. So it's the genetics of how you metabolize drugs. And it's a very, it's really one of the most advanced aspects of what we call genetic medicine.
So what are you working on currently there, Doctor? Where do you spend most of your time?
So much of my work is in the clinic, but the clinics that I'm holding now are very different than the clinics I would have had 10 years ago. So we are now, for example, in the past, I would see kids with birth defects, and people would want to understand what caused these birth defects. Was there an underlying genetic change? Because remember, that's the instructions for how a person's supposed to be put together. Now, we're getting referrals from physicians who are saying, I have a patient who has depression and I've tried them on every different medication and nothing is working. We need you to help us understand their genetics and what drug is going to work for them.
Okay, so for all the changes, and you said in the last 10 years, how does that affect you as a professional working, behind the scenes with all of this then? I would imagine that's been kind of a crazy decade, huh?
Yes. No, so our practice has changed very quickly.
Oh.
We've had to be very flexible. We are taking in a lot of new information. The technology is generating a lot of new information that only specialists can really analyze appropriately and put in context for the patient. And more recently, with the democratization of genetic testing, direct-to-consumer testing, for example, our practice is continuing to shift and we're having to adjust accordingly.
Yeah, let's talk about consumer testing just a little bit and the DNA testing kits. This thing, it's become so popular. Tell us just about the kits themselves.
Sure. So, I think this area probably started more in the science of ancestry and understanding genetic ancestry. And so most of the kits that you see out there focus on, or at least have some component that looks at a person's ancestry. And the way they do that is there are a number of databases out there that have analyzed genetic material from people of different racial and ethnic backgrounds. So when a person gets analyzed, they look at their DNA and they compare them to those different ancestries and try to make a prediction as to what percentage might have come from the different groups. Over time, people started saying, well, maybe we can do more than ancestry because we know there's a lot of health traits that are encoded in your DNA, and can we provide some information to people about their particular risks for certain conditions. One of the things that you have to keep in mind, though, is that the way that they're currently making risk predictions is by doing, they're looking at what are called association studies. So just to step back for a minute, say we're focusing on diabetes and we want to understand the genetics of diabetes. We take 1,000 individuals with diabetes and we analyze their DNA, and we analyze the same DNA from people who don't have diabetes. And we look to see what are the differences. What DNA sequences are more common in the diabetes group versus the non-diabetes group? Now, those sequences don't necessarily cause the diabetes. They're simply associated. Okay? They're merely just sort of more common in that particular population for whatever reason. And we don't fully understand that relationship. Some of the companies, what they'll do is they'll take that information from those association studies and they'll flip it back. So for example, if they test you and they find that you have one of those changes that's more common in the diabetic population, they try to convert that into a risk number. And it's challenging to do that because we don't really understand the relationship between that change and the disorder. And so some of those risks that they're providing is, you kind of have to take it with a grain of salt.
Okay. Now, these kits, right? Pretty simple. What do they involve and how do they work?
So the first thing you have to keep in mind is each kit tests for different things. So you're going to want to know what is it that you're trying to get information about.
Okay.
So look to see if it's if it's focused on an ancestry kit because that's what you're interested in. If it's a health kit, what is it looking at? There are kits out there that look at your pharmacogenetics, which I mentioned before, your ability to metabolize certain drugs. There are kits that will try to predict what diets might help you best for weight loss. There are kits that will look at what's a good nutritional program for an individual. Some of those probably aren't as scientifically validated, so you just have to keep that in mind. But the actual process of the kit is very simple. So DNA is available pretty much from any part of your body. The most readily available is actually within the saliva or within the cheeks. And so many of these kits are simply tubes that you have to spit into to a certain level. And then you just close the top and you twist it, and it allows a preservative to preserve the DNA. And then you pop it back in and put it in the mail. They extract the DNA, do the analysis, and then usually send you an email link to a portal that you can log into and begin to look at your DNA information.
Fascinating. Okay, there are a lot of kits, and you talked about, folks wanting to know specifically, but with all the kits out there, how do you know where to even start?
Right. So what I would say is, for a person who's interested in ancestry, there's a lot of options out there. Your best bet is going to be a kit that has a database of a lot of individuals in it. So the more popular kits are going to probably have more accurate information. So Ancestry, for example, or 23andMe probably have the most samples that are in their system that they can sort of give more accurate, slightly more accurate information. You have to just keep in mind, though, that human genetics is still a field that's evolving. So we can't accurately tell a person, you are 10% from this particular county in this particular country of Northern Europe.
Yeah.
It's merely saying that your DNA resembles people from this area more than it does anybody else.
And I wanted to ask you about the accuracy, because I think people they watch the commercials, they take the test, they get all excited, they send it in, and then it comes back and they're like, wait a minute, that didn't narrow the scope nearly as much as I had wanted it to.
Exactly.
So I'm glad that you bring that up. Are there things that people learn that maybe they weren't expecting to learn when these results come back?
Sure. So one of the things that often happens is people know their family history, or believe they know their family history and their ancestry, and do the test. For example, they believe they're of Italian descent, and then there's no Italian that comes up in the ancestry. And that can be for a number of reasons. It could be that the particular Italian group they came from is not well represented in the database. So if they're from a very tiny town in Italy and no one from that town has ever been tested, then it's not going to pick up that you have Italian ancestry as a simple issue.
That's a good point.
Another issue that can potentially come up, and has come up, is non-paternity, meaning the person who you thought was your father wasn't in fact your father. And that can be discovered, of course, through genetic testing if your parents are also tested.
Okay. Anything about these home kits that I haven't asked that you think it's important for viewers to know? Because this really has become a pretty hot topic. If you're not getting one for Christmas, you're probably going to get it for somebody. So anything that we need to know that we haven't addressed?
What I would say is if a person has a family history of a particular condition that has a genetic association, for example, if you have a strong family history of breast or ovarian cancer or a strong family history of colon cancer, the testing you want to do is a clinical test. You do not want to use a direct-to-consumer test for that analysis. And there's a number of reasons for that. Number one, the stringency that has to be maintained in the laboratory is far more rigorous in a clinical lab. Number two, the type of testing and the type of genes that we would look for will differ depending on your particular family history, instead of just sort of a blanket test that everyone is getting. So in those instances, I would suggest a person seek a geneticist or a genetic counselor to review their family history and to say, your family history looks like maybe one of these 10 genes, and we'll do a test that will look for those particular genes.
Very good advice, thank you. Okay, Jason, we're going to change gears. We're going to go from Ancestry.com, 23andMe, to the crime lab, right? So before we do anything else, paint a picture for us of your crime lab.
Our crime lab is a full-service crime lab, and we serve the entire state of Nebraska. It's a for-free service for any law enforcement agency in the state. It's a tightly controlled facility because of the work we do, but it's also a very diverse facility. Not only do we do DNA testing at the laboratory, we also analyze seized drugs, for example, the stops you see in the news along the interstate when they seize the drugs. Those are tested at our facility. Toxicology testing, trace testing, arson, for example, looking for accelerants. We have specialists that do fingerprints, that also look at firearms and tool marks in addition to the DNA. So we're a very diverse laboratory where we do applied science.
How many people are we talking then, Jason, in all of that?
Sure. So our laboratory has, I think, approximately 32 people that are full-time there.
Okay. And your role is what?
So I oversee the DNA testing at the laboratory, ensuring that we're using current and proper methods for the DNA testing that is done on biological evidence.
Okay, and you became involved. I asked the doctor too, how did you become involved in all of this?
Well, I was one of the unique people that never changed my major in college.
Wow.
But I had the opportunity to tour the crime laboratory. This was before CSI was so popular. And became very interested in forensic DNA testing at that time. And just managed to fall into that career.
Okay, now we have a video that we want to show our viewers, a little bit of history from the crime lab standpoint. So we're going to go ahead and roll that, and then we'll talk a little more when we come back.
Constable Baker is certain Richard Buckland killed both Linda Mann and Dawn Ashworth. Struggling to make a case, Baker has an unusual idea.
I was aware at that time that Dr. Alec Jeffries had been working at Leicester University in respect of DNA tests.
Dr. Jeffries knew human DNA is mostly identical.
And well over 99% of our DNA is the same between people. That allows us to have 2 ears, 2 eyes, a nose, 4 fingers and a thumb on each hand.
But Jeffries wanted to know more about that other 1%. In 1984, he discovered a patch of DNA that was unique to each individual, a sort of DNA fingerprint. And when Constable Baker thinks about Jeffries' work, he has a question. If DNA can prove paternity, can it also prove whether Richard Buckland killed Dawn Ashworth and Linda Mann? The police are willing to try anything. They give the lab the crime scene samples. These researchers may know DNA, but they've never used it to solve a crime. The results of this real-world experiment are anybody's guess.
Okay, so they were motivated to try it. How does it work?
So as has already been mentioned, the DNA in our body is the same no matter where it comes from. It's in the nucleus of the cell. So if we take anything that's left behind from a crime scene, anything we touch, we leave part of ourselves behind. So if I'm holding a cup, for example, I'm leaving skin cells behind on that. So whether it be a blood sample, a saliva sample, or even just skin cells that are sloughed from our hands, we're leaving our genetic material behind. So we can collect that, extract the DNA, do a DNA type, and then compare that to known samples from victims, suspects, and elimination folks.
Jason, you've been doing this a long time too. What have been some of the greatest advances that you've seen in your career?
Probably in the area of the sensitivity of our testing. Theoretically now we can get a DNA profile from a single skin cell. So the sensitivity is very high. But along with that come a lot of challenges. One is contamination, but even more than that is, what does the DNA profile mean? Just because we obtain it, what does it mean? Because DNA from just a few cells could be explained a number of ways. If I shake your hand and then shake somebody else's hand and they walk out the door, theoretically, we could find your skin cell on the doorknob that you never touched. So just finding your DNA there by itself has to be put into context.
This is really very fascinating. All right, so most interesting case. I would imagine you've worked on a lot of cases in your day. I won't even ask you for a number. But based on the discussion we're having today, is there one in particular that you find interesting enough to share with our viewers?
Well, there's one case that comes to mind. With the technology we're using currently in the crime laboratory, we cannot distinguish between identical twins. So I have had a case that involved identical twins where one was a victim and the other was not. Probably the most interesting case I was ever involved in, however, was what was thought to be unidentified human remains, and we could not obtain a human DNA profile from it. And while it was never confirmed, we're pretty sure it was actually the paw from a bear.
Really? And I suppose that brings up a whole different thing, right? Especially in your line of work where you're pursuing something, you're thinking it's something, and then it turns out to be something completely different. Do you run into that a lot?
Well, we try to be very independent at the laboratory and not focus on the crime itself. We just are there to answer the questions, so we try to remain as unbiased as possible.
Okay, objective, right?
Correct.
Yeah. So as we look forward, especially in your line of work, Jason, what are we seeing down the road, do you think, in terms of advances or changes to help make what you're doing a little bit easier? Sure.
A lot of the big changes that are occurring right now are in the area of software. So we deal a lot with DNA mixtures. So we get mixtures of multiple people's DNA in one sample. If 2 or 3 people were bleeding at a crime scene and their DNA is mixed, we have to be able to sort out those mixtures, whose DNA is whose. And so we're really moving into this, using software to assist us with that. Additionally, the community is slowly moving to the full sequencing process that Dr. Rahman talked about. Because of legal challenges, those technologies that are used in other disciplines are a lot slower to be implemented in the forensic science community because they have to overcome legal challenges.
I'm listening to the two of you, and I would imagine it takes a unique person to do what you are doing. Jason, what characteristics do you think? I mean, I'm sure we've got some folks out there who are fascinated with what's going on, and they want to do what you're doing when they grow up, or maybe in their next life. What do you really need to possess to be able to do what you're doing?
One is to be very detail-oriented, and I would say most everyone at the laboratory is very type A personality and very dedicated to what they do.
Okay, thank you. Dr. Rahman, the future of genetic research in general, you've been doing this a while as well, and so what are you seeing down the road?
I think what most institutions are looking at now is how do we integrate genetic information in your routine healthcare. So, for example, it may be in the future that you've had your entire genome sequenced and it lives as part of your medical record in the background. And when you show up with a complaint to your physician, say something is hurting you, or you're experiencing some unusual symptoms, that the first thing they do is they just call quickly through your genetic material to see, is there anything there that might explain those symptoms? And then if they choose to prescribe something, once again, they use your genetic information to give you targeted therapy. I think that's where we're really going, and I think the quality of care can really be improved. The speed to a diagnosis will be much faster.
Amazing. Well, we have some websites that we want to put up for our viewers. We're talking about DNA testing today. So if folks want more information, they can go to unmc.edu/MMI or statepatrol.nebraska.gov. Gentlemen, thank you so much for being here. This was one of the fastest half hours I think I've ever been a part of. Fascinating stuff. Keep doing what you're doing. Best wishes moving forward.
Glad to be here. Thank you.
All right. And thanks to our students for working so hard behind the scenes. Of course, to our viewers for watching. As we close, a look at the Joslin Art Museum as seen through the lens of Ryan Gerchen, a senior here at UNO. We'll see you next week.
Consider This is an issues and information program produced by UNO Television. More information and program archives can be found at netnebraska.org/considerthis. If you'd like to share your comments about this program, call UNO Television at 402-554-2516 during regular business hours, or send an email to cwyatt@unomaha.edu. Support for Consider This is provided by
There's a new place for senior care, a place with flexible meal plans and 24-hour room service. A place where seniors get the care they need in the comfort of home. Home Instead Senior Care.